All right, everyone, here's notebook page 7, Biology and Behavior for AP Psychology. You can take down the title card now. Let's go to slide 1, unconscious information. So when you think of how our brains process information, think of it like a computer processing millions of bytes of information every second. But that processing split in our brain. We have the conscious and the subconscious or unconscious mind. This is called dual processing. Both sides are processing at the same time. Our conscious processing is roughly limited to about 40 bytes, so not very much. It's defined of what we can analyze, what we can consciously think about. When you sit there and think of something, that's your conscious mind. But our subconscious or unconscious is processing the remaining 11 million bytes. That's an approximation of the percentages there, but that's devoted to that unconscious processing, the sensing and perceiving of stimuli, what you smell, what you taste, what you touch. This is our automatic sensation and perception of the environment, the objects that you're seeing, their distances, the texture, the movement of things. So, I can't even put it into words, right? For you, if you would have to consciously process seeing movement, you couldn't do it, right? There wouldn't be a possibility of that. So it has to be subconscious. So, for example, just kind of an example to show that, if I showed you a 5-second clip of an unknown high school basketball team, told you to focus on the basketball, and you did that, you wouldn't remember the team colors, you wouldn't remember the players' numbers, you wouldn't remember what the cheerleaders were doing. But if they were using an old ABA-style basketball that was red and white, you'd be able to describe that basketball. You'd probably be able to describe the brand. You might be able to say what happened in terms of scoring baskets, right? But that's your conscious mind kind of working, but you saw everything else, So your unconscious mind has that information. So let me put up the slide here. Write this down in your own words. Let's move on to slide 2. Frontal lobe. We will finally get to our boy Phineas Gage here that we started the unit with. And most of our conscious activity occurs in the frontal lobe, the prefrontal cortex, that cortical region of our brain. So this is the last area of your brain to fully develop and connect to everything else. So most of our creativity, most of our problem solving, most of our abstract thinking, our judgment, impulse control, personality, that's in the prefrontal cortex. And so that frontal lobe, same thing. It doesn't deal with the biological processes of the brain, so that's why it can be the last one to fully develop. If the frontal lobe processed our breathing, if it was the last thing to develop, we'd all die. So this doesn't develop fully until your early to mid 20s. So when we talk about adulthood and turning 18, some of you might be turning 18, you're adults. Your frontal lobe isn't fully developed. That creativity, that problem solving, that recognizing what consequences might happen. So it's something. The impulse control, for example, would stop you from punching someone in the face who's really annoying you. And there'll be other examples on the slide, but that's something that doesn't develop. So some of those choices. That's why we see in college, especially young college students, on their own, that frontal lobe is still not fully developed, and sometimes some of those choices and consequences doesn't quite mix. So, and Phineas Gage is a great example of the impact on personality, on impulse control. We still use that case study as an example. So I'll be right about him in these notes as well, so you have it down. I'll put them up. And you can put them down at this time. Move on to association areas. So we can map the brain by probing it, which is electrical stimulation. So we can see what's happening in the brain and getting a response from each of these areas. We can see when we get responses and when we don't. One of the areas that does not get a response is the cerebrum. And the cerebrum is responsible for your thoughts, for your memories, for problem solving, for personality. So it's part of that frontal lobe. And part of this is the association areas that interpret, integrate, and act on sensory information, linking it with memories. So, your senses, the things you touch, smell, taste, can be linked to memories. This happens a lot for me. I wore the cologne Curve in high school. And so if I ever smell that, it's very rare that I smell that cologne. But if I smell that cologne, it really takes me back to high school. I remember my senior prom, and I remember some of these memories that I don't think about are associated with that. And another one for me is like certain car air fresheners that I know there were cars in my childhood that my brother had or my parents had that had certain air fresheners in there, and if I smell that again, it takes me right back to elementary school or middle school. That's what the cerebrum does. So another reason that children often have inferior fluid, crystallized intelligence, in their association areas, is they've been saturated with fewer memories, experiences, and skills than adults. So this doesn't happen for younger children because they don't have the memories to associate. They don't have that association areas yet. So this is called a working memory. We learn more efficiently using the knowledge we've acquired and built upon as we age and as we learn. Building on those past experiences helps us learn more. So, for example, you can't spell a word without having a foundational working memory of letters. You can't calculate math problems without a basic working memory of numbers. Storage of information isn't in one specific area of the brain. It's actually different for everyone, something that we found with that probing of the brain, which makes it difficult. We actually can store information in different parts of the brain, and that works very differently. So let me put up this slide and write this down, and then we'll continue talking about this for a bit. So association areas are also responsible for recognizing objects and faces. So these are called feature receptors in the brain. So you might recognize a face a decade later, but not know the name, because it's much easier to recognize that face because we have a specific area for recognizing those faces. Those feature receptors are going to look at that face. They're going to recognize that face. And so, if you've ever seen this, to me, this happens a lot with actors, right? Watch a TV show, watch a movie, and I'm like, I know that person. Like, I don't know what I've seen them in. That happens all the time, But if you had said their name to me, that maybe I came across once or saw in the credits once, I wouldn't. I have no idea who you're talking about. But if it's someone, not someone very famous, but if that face, I recognize it. I saw it, 8 years ago in this 2 episodes of this one TV show. So that is those feature receptors. And if it's damaged, you actually may not recognize faces. This is called prosopagnosia, and it's something that if that area of the brain's damaged, people won't recognize faces anymore, which would be very, very strange and problematic. So this up. If you want slide 5, hemispheres. So our brain is more similar to kind of 2 brains that communicate with each other, as shown by the research by Michael Gazzaniga and Roger Sperry. And we'll probably look at some of the research later. But using the corpus callosum, which connects the 2 sides, the left brain and the right brain, you see in the picture up here, the 2 hemispheres coordinate and communicate almost instantaneously. So, roughly the left side kind of handles the mastery of the known. So things like language, mastering language. The right side, more the unknown, creativity, problem solving, etc. This is hemispheric specialization. That some functions are more dominant in one side of the brain than the other. Now, this being said, people are not right-brained or left-brained. That's one of the myths that we expelled at the beginning of the course. That's not true, that you actually use both sides for every task, but there might be a leaning toward one side in terms of that hemispheric specialization that happens. It's likely been developed through evolution to analyze new information that we need to survive, because we need to use both for survival. So we use both. You're not merely one side or the other. So we'll talk a little bit more about split brain research in these notes. Let me put up this slide, and I'll let you take this down at this time. So the motor and sensory neurons are connected to the brain laterally. So each side of the brain controls movement, visual fields, etc. of the other side of the body. So the left side of your brain controls the right side of your movement. The right side of your brain controls the left side of your movement. This is called brain lateralization. So the left eye's information is only received by the right hemisphere. It is communicated to the left hemisphere only due to the operation of the corpus callosum. So, the only way that we can use both sides of our body in tandem is if that corpus callosum is there and persistent and works well. So, all right, my apologies. There is the hemispheres continued. Write this down now. Let's move on to split brain research. And so the Gazzaniga and Spiri experiments, like I said, which we'll get into later, show that if that corpus callosum was severed, you would not be able to use language to express what your left eye sees when your right eye is covered. However, you could point to the word or answer with your left hand, but not your right hand. So pretty crazy, right? So if you cover your right eye, you could see something, and because of that right brain function, then you couldn't use language, but you could point to it with the other side. It's very interesting to see. So it's something that we'll get into likely today. Another example, the right side tells your body to walk, but if I sever that corpus callosum, the left side doesn't know why you're walking. So, it may think that, hey, you're going to get a Coke, even if you're trying to leave the room. So it doesn't know why that's happening. So very interesting stuff here. I'll let you rate this short slide down. Let's move on to slide 8, repair. The brain repairs damage in two ways. Plasticity, which is reorganizing of existing neurons and neural connections, with the slower and more rare development of new neurons called neurogenesis. So plasticity is usually how the brain is repaired. Neurogenesis is very rare cases that can happen. In either case, once a neural connection is completely severed, if there's damage that completely severs that neural connection, it cannot be repaired. The brain and the nervous system can try to develop or repurpose other neurons or connections to make it work. So, and that's what plasticity really is. That's really what will happen. So, for example, if you have brain damage that causes the loss of your limbs, sometimes the brain can be reprogrammed to work properly again. If I can't use my left arm, then maybe my neurons will be repurposed for that movement from other movement. So plasticity can only use similar neurons to achieve this. Auditory neurons, for example, can be repurposed for other senses, things like that. So we'll get a little more into it in the next slide here, but I'll put this one up so you can get the definitions down. It's going to move on to repair continued. The brain can compensate as well. So the loss of sight or hearing, blind, deaf people can focus more on their remaining senses. It's not that these senses are like super senses, but that focus can lead your brain, that conscious processing power, right? Those 40 bytes that we're processing can be focused more than if you had an additional sense. So, much like a computer with too many programs, tabs open, processing speed suffers. And this happens with your senses too. We have so many senses that we have to try to focus on all of them, and the processing suffers. So if someone's blind, those neurons can be repurposed for other senses. So that's the example I was kind of getting at there. If you don't have sight, those sight neurons can be repurposed for hearing. That's usually what happens. Someone lost their left arm, their right arm would work better because those neurons would be repurposed for their right arm. So we've seen this in studies with soldiers that have lost limbs in war and adjusting to working with one arm, with one leg, how those can work better. So for someone who's blind, it would be reading Braille, maybe the touch. For someone who's deaf, using sign language, that type of interpretation as well. So this slide, I'll give you a little bit to write this down. Move on to slide 10, lesioning. So the Broca's area right here controls language expression. This was discovered by Paul Broca, so it's named after him. And damage to it causes aphasia, the ability to understand and formulate language. So if there are lesions on parts of the brain, like on that Broca's area, you can't formulate language. You can't speak. You can't. It's not that you can't talk. It's not that you're that doesn't work, but it's that you can't communicate with language that you know. So this was found in the autopsy of patients that they didn't know why they were just not making sense ever, and that's what they found. So and this works for other areas as well. I mentioned my brother who had brain cancer, and depending on where his kind of lesions and tumors were, and what they were putting pressure on, depended on what was happening in his body. He had pressure on the area that made his right arm not work as well. It's very difficult. There was some pressure on the Broca's area. So he had really trouble like thinking, processing language, and formulating that language. So it all depends. When lesions occur, when tumors occur, that pressure they put on can have an effect. And then the Wernicke's area, which was discovered by Carl Wernicke, controls language reception, plays a key role in language development. This is kind of how we perceive language and then develop it, something we'll talk about later in the course. So let me put up this slide, and you can take it down now. Slide 11. So we'll talk about genetic and environmental factors on behavior. This is going to be just some different factors on behavior, and something that we'll get into later in the course as well in more detail. But if you've seen the background of these slides, have been a pair of twins, right? That are both astronauts, and so we'll get into that at some point here. So here's slide 11, evolutionary development. So like the Charles Darwin theory of natural selection, there are traits that allow populations to reproduce and survive from generation to generation. We've talked about some of those, the phobias of snakes and heights and things like that. So that's an example of those traits. Now, things like the evolutionary bonding mechanisms between mother and infant, oxytocin, which I think you did an assignment earlier, perhaps, or will do an assignment on, on that chemical. That's something that's an evolutionary chemical that works. So that allows for that bonding to happen. Hey, this is my mother, this is my caregiver, right? And this is something you can be bonded to. Another example of evolutionary development is sex, biological issues of sex. For males, there's a drive for multiple partners because males can do that, right? And this need for reproduction is can be different. Some we'll get into later in the course can be different. can be different genetically. So a need for social contact, all these things are kind of evolutionary traits. So something we'll get into later, but know that, hey, evolution, theory of natural selection, there can be things that are just passed on from generation to generation that isn't necessarily a development of, who we are just from our personalities. We'll put that up here. Let's move on to genetics. But personality, and we'll have kind of a part of a unit on personality itself. We'll go in more detail. A person's character, their interests, their temperament is largely genetic. And how do we know this? We've found this out in a couple different ways. Adoption studies have been a big one. So when we've had a child who's been adopted to a different family, and then we've assessed their personality compared to their biological parents and compared to their adoptive parents, they've reflected the personality of their biological parents, people they've never met. And that's because genetics plays a big role in that personality. Identical twin studies have been a big one that shows a role in genetics. There are some rare instances where identical twins have been split up at birth and have been reintroduced. And there's a couple scientists, that's all they do. They travel around the world when this happens, and they study these pairs of twins. And so we've seen, since they're nearly genetically identical, and that's what identical twins are. They're genetically identical almost, and fraternal twins are just like siblings. They have the same genetics as siblings. So we've seen these identical twins have similar personalities, have similar preferences. And I'll show you a video of a TED Talk about this topic. That's really interesting. One of the lead researchers in this. Hormones play a major role, right? Particularly sex hormones, testosterone, estrogen. Those are regulated genetically, and so that's something that occurs. As I mentioned before, sex drive, functions are generally genetic. And then this deals with that evolutionary trait as well. These can be linked together. Males tend to be more motivated to pair more with females with little disregard for the quote unquote quality of the female, the choice of the female, because that kind of the evolutionary trait of reproduction, a male can impregnate multiple females, and that's kind of the goal. Whereas females, it's going to take you 9 months to have a baby, to reproduce, and so there's more choice. And so even with sociocultural influence, even with our culture saying, hey, that's a double standard, that's not right, and it's true, right? That that you kind of have this double standard of men can go out and have sex with as many women as they want, and women shouldn't. Even that kind of double standard exists. And so even with that, that influence that happens, or if there's some cultures where that's flipped a little bit, that still ends up being the case. So kind of interesting, some of the studies that have been done on that, and some we'll get into later in the course as well. All right, let's move on to the final slide, 13, which is environment. And so environment can impact personality and behavior, aside from not developing properly. So if you're raised in an unsafe environment, if you are raised in you have malnutrition, you don't have enough to eat, not being educated, etc., environment can still affect biology. It can affect genes. So all those things can have a big effect. Even if those things don't happen, your environment will still affect you, right? So in interaction, genetic changes occur due to environmental stresses, right? Factors. Excuse me, I was jumping the gun, because stress is a big environmental factor that can affect your genes. If you're under constant stress, your genes will change. Illness, exposure to chemicals, things like this. We know that, hey, that's your environment, and it can turn certain alleles on or off to change your gene expression. This is called epigenetics, that the environment will turn off certain alleles and change how your genes are expressed. So how do other things affect it? Parenting and how you're raised. We'll get into that later. But this is really bad. Environmental issues can the things year round can have an effect. So let's write this down, and that is the end of these notes. Thank you so much, and be good.